This is the virtual lab for antibiotic susceptibility testing. This is a big thing that happens in clinical labs. What we're trying to figure out when we do this is what antibiotics can the clinic use to treat a bacterial infection. So if a patient has a bacterial infection and you manage to isolate the bacteria that are causing that infection, what antibiotic can you use to treat them? Now, in a lot of cases, clinicians see the same kind of infections again and again. They see a lot of staph and strep, and they know what antibiotics to try first, and that usually works. But in other cases, that doesn't work, and the clinic would want to get a real answer about what would work in this unique case. So that's what I'm going to show you. There are two real things that control this. One is what antibiotics are it susceptible to, and the other is what dose would be required. So sometimes we're thinking about using doses that are impractical. So in a lab, we can look at the concentration of an antibiotic that would kill it, but in the body it might be very difficult to achieve that concentration in any particular tissue. So, for example, what concentration will you find in the brain? For a lot of antibiotics that don't cross the blood-brain barrier, the concentration would never be high enough to treat an infection. And that's the thing that we can see with meningitis. You need to use an antibiotic that can cross that barrier. And so that's the sort of thing you'd think about. But once you know which antibiotics will go where, then you need the other data, which is which antibiotics and what concentrations are necessary. So I'm going to show you two things. I'm going to show you the minimum inhibitory concentration. This is a direct measurement of how sensitive bacteria is to an antibiotic. And I'm going to show you disk diffusion, which is how we figure out which antibiotics to use. So minimum inhibitory concentration is a very precise number we get that tells us for a given antibiotic and a given bacterium what concentration of the antibiotic is necessary to stop that bacterium from growing. So remember, some bacteria are some antibiotics are bactericidal, so they kill the cells. Others are bacteriostatic, so they stop them from growing. And in either case, they can work, but I can't say what concentration kills the cells because a lot of antibiotics don't kill the cells. So instead, we say what concentration inhibits growth, and that's what we're looking at here. So what you see here are culture tubes, and what they did is they mixed up some growth medium with all the nutrients the bacterium might need, and they added some antibiotic to it, to this first one, and then they did a serial dilution of that antibiotic. Instead of the 10-fold dilutions I showed you earlier, they do a 2-fold dilution. So this one is half the concentration of this one is half the concentration of this one is half the concentration of this, and on down the line. And what you end up seeing is you can still cover a few orders of magnitude this way. Roughly every 3 dilutions is roughly a factor of 10. So if you have 6 dilutions, that's roughly a factor of 100. We also have a control tube where we don't have any antibiotics at all. And the idea is, in this tube, we expect bacteria to grow. We are going to put our mystery bacteria from the patient in this tube, and they should grow. Also, we'll put them in this tube, and chances are this concentration of antibiotic is too small to affect them, so we expect them to grow. And so what we do is we look at what concentration do the cells grow, and in this case, this color indicates growth. So in which tube is there growth? Well, there's growth all the way to this one, and then here there's no growth. So we would say that the minimum inhibitory concentration is 6.4 micrograms per milliliter. Now that's not something you can immediately use to drive a dosage. That's not something off the top of your head you could then use to give to a patient. But you could look up in a table what dose is going to give you what concentration in a particular tissue. And then you'd have to compare that to your individual patient's health and whatever's going on with their physiology, and it would get really complicated. So this is the basic piece of information we have about one antibiotic and one bacterium of interest. And this is the minimum inhibitory concentration, or the MIC. And so this is going to come back towards the end of the lecture. So make sure you understand this, pause the video, go back if you have to. The next thing we will look at is antibiotic susceptibility. So the minimum inhibitory concentration is a good thing to know, but which antibiotic do you test? We want to use antibiotics, but remember there are a lot of situations where bacteria are resistant to antibiotics. Maybe they have a plasmid that gives them an efflux pump that they use to push the antibiotics out of the cell. Maybe they have a beta-lactamase enzyme that works against the penicillins but not the cephalosporins. Maybe they have an extended spectrum beta-lactamase enzyme that inactivates all the beta-lactams. We don't know until we test it. And so this is showing how you might test it. And so if we can isolate an organism and grow it in the laboratory, we can figure out what antibiotics might work against it. So the technique we use is called a disc diffusion assay. And again, there are others you could do, but this is one that we would have done in a laboratory setting in a class. This is the simplest way to answer these questions, and it is absolutely valid. You could use this in a clinical laboratory. So you might see this called the Kirby-Bauer method, and that's a very specific technique using specific recipes. And this is more generalized version. This is a specific example. Okay, what do you do in this assay? First, you take a petri dish with media on it, and you spread bacteria all across the surface. You cover the whole surface of the petri dish with a small number of bacteria so that when you incubate them, they will grow all over the surface of the plate. That's what you're looking for. Immediately after you put the bacteria on, you add antibiotic saturated discs. So these little discs that have known amounts of antibiotics, you put them on the surface, and the idea is the antibiotics will diffuse out from the discs into the agar. And then you incubate the plates and see if the cells will grow. And you analyze your results. So let's look at it. Let's apply bacteria to the surface. So I'm just going to kind of cover the whole surface. And it doesn't have to be beautiful. Like I can, if I want to squiggle, it really doesn't matter. There are a lot of different ways to do this. I've never seen a professional do squiggles like this, and that would kind of be stupid, but it doesn't matter. The point is, as long as you cover most of the plate, it's enough. If we incubated this plate, we would expect to see cells grow wherever I put them. And I obviously didn't do a great job of putting the cells on there, but wherever they are, we kind of see the line thicken as cells sort of colonize and spread and grow. Where there was one cell overnight, there will be 1 million cells, something like that. So this is what we would see. So let's go back and just think about, we inoculate our plate and then we put antibiotic discs on it. So they're hard to see, but these little discs would be little paper discs that have to be sterile. And we put the sterile paper discs on there, and they have known quantities of antibiotics in them. And so the antibiotics will diffuse out from the discs into the agar in every direction. And what that will do is set up concentration gradients where right around the disc there's always going to be a lot of antibiotic, and the farther away you get, the less antibiotic there will be. And so you'll reach a region far away from the disc where there just isn't enough to inhibit any bacterial growth, and that will matter in a second. We'll see that. So if you then incubate the plates, what you start to see is that cells can grow in some areas but not in others. And so in some places, the antibiotic concentrations are high enough that the cells can't grow. This is what it would actually look like in a laboratory. You'd have a much nicer pattern, or you'd use a liquid to evenly coat the plate with bacteria before you incubate them. But either way, you'd see these nice circular zones of inhibition. And what you're seeing is bacteria grew here. They did not grow here. You're seeing through the agar here, and there's no bacteria. And it's all based on the fact that whatever these cells are cannot grow if there's this antibiotic in this concentration. But when you get far enough away, that antibiotic is dilute enough that the cells can grow. And so what we do is we measure our own zones of inhibition. We look at the diameter of our zones of inhibition, and we can compare them to tables that have been published. Because researchers have found for each of these antibiotics, they have found that whenever you're trying to treat a certain species of bacteria with this antibiotic, it fails if you get a certain diameter, and it would work if you got a certain other diameter. So let's look at that. So we're going to look at the table. This slide shows you instructions for looking at the table, but let's just go there now. So let's start with the example of erythromycin. Let's just take a look at erythromycin. So let's see what we have. Erythromycin, 30 millimeters. That's that distance, 30 millimeters. So we compare it to these numbers, and what we see is 30 is bigger than 18. So 30 goes here. The other number they get is 13. Well, 30 is much bigger than that. So 30 is over here, and that means our mystery cells are completely susceptible to erythromycin. So erythromycin would be a decent choice for treating this infection. So let's look at another one. Let's see. Oh, let's look at streptomycin. This one has a really small zone. So what that means is the cells need to have a really high concentration if they're going to die or stop growing. But if there's a moderate concentration, they're just fine. So the concentration you need to use to treat this bacteria with streptomycin might be pretty high. We'll see. So the diameter we got is 13 millimeters. And for streptomycin, susceptible cells would be over 15. So we would need to be 16 or higher to call it susceptible. For it to be a good treatment option, we would have to have a diameter of 16 millimeters, and we don't. Instead, we have 13. Well, 13 is still higher than this resistance threshold. 13 is in between this susceptible and resistant. That's an interesting case because that means it's intermediate. That means we might be able to use that antibiotic in an emergency, but we would have to use a really high dose of it, a higher dose than we typically want to use. So that would be intermediate. The last topic I want to mention is just what governs the size of those zones. Well, the microorganism, like what is the organism and how sensitive is it to the antibiotics? That's the main thing. That's what we care about, and that does determine the size of the zones. And it's going to be different for every isolate of every bacterium, every species. So we do the best we can. And for common pathogens, we have the data to know what it means if there's a certain size of zone of clearing. That was an awkward way of saying that table on slide 16, that table is a small subset of a giant, many pages long table you can get that would guide you in this case. Well, another thing that controls the size is how fast the antibiotic diffuses through the agar, because big molecules diffuse slower than large molecules do. That's physics, and it's complicated. And so we can't go out and predict how big a zone would have to be. Instead, we just measure this empirically. And anyway, that's why if you look at the table, that's why you need a different entry for each one. It's just the different properties of these antibiotics are going to determine how they diffuse and how susceptible bacteria tend to be to them. And then the last one is you can get antibiotic discs with different masses on them. And so the antibiotic and the mass of total antibiotic on the disc, they're both printed on the disc. So for an example, you can get tetracycline. I think you can get a 10 microgram tetracycline. I think you can get like a 10 milligram tetracycline disc and a 5 milligram tetracycline disc. And that would determine what, to some extent, the size of the zone of inhibition. That's enough for zones of inhibition and minimum inhibitory concentration, and I hope after looking through this and working through this, you will have a sense of what might be going on in the clinical labs in the basement of a hospital.